I'd like to talk a little bit about quality control of medicinal mushroom products, and that's something that has been an issue for the last 40 years. What can we do to guarantee higher quality? This has been going on with mushroom scientists, and they've all been talking about how we could standardize on the basis of one or two of the main active constituents. There are many different fungal parts that are sold as medicinal mushrooms. Mycelium is one of them, and pure mycelium is actually made in liquid fermentation in China. The most famous of these products is Cordyceps CS4. In the United States, companies will grow mycelium on grain, what is a process actually that was designed to manufacture grain spawn, which is the seed for growing mushrooms. Mushrooms are the standard that have been used for thousands of years in the medicinal mushroom industry and traditional Chinese medicine. Sclerotia are hardened masses of mycelia, which generally speaking grow underground. A good example of this is Wolfoporia extensa, or also known as Poria cocos. Chaga, which oftentimes is referred to as a sclerotia, is actually a canker, a manifestation of a fungal disease or infection, often of birch trees. Chaga has very low amounts of mycelium. So at Nammex, we've developed an analytical fingerprint based around 5 different markers. And again, these are markers, quality control markers for us. The 1316 beta-D glucans, Agosterol, triterpenoids, ergothioneine, and alpha-glucans. We use the Megazone Test for our beta 1316 glucan testing. This test does not measure grain beta-glucans like oats. It also measures alpha-glucans. It's accurate and repeatable. It's been peer-reviewed and validated. And beta-glucans make up 50% of the cell wall of a mushroom or mycelium and absolutely provide a clear fingerprint for each species. Lack of beta-glucans will basically indicate a lack of fungal tissue. And mushrooms do not contain starch. They have small amounts of glycogen. Starch that we pick up generally indicates the presence of an adulterant or other materials such as residual grains. The Megazone test was published in the peer-reviewed Journal of AOAC International in 2016, so it is definitely a validated method. In 2016, I presented my analytical fingerprinting method to the International Society for Mushroom Science. And what I found in my analyses was that mushrooms contain from 25 to 66% beta-glucan and very low alpha, from 0.1 to approximately 6%. The exact opposite I found in myceliated grain products, where the alpha-glucan was 24 to 70%, beta-glucan was 0 to 13%, so it's just the opposite of what you'd expect to find in a medicinal mushroom. Regarding Agosterol, which is a very good indicator of fungal material, I found that mushrooms in general contained around 0.1, whereas these different mycelium products contained about 1/10 that amount. In 2019, after analyzing many more samples, and in this case 167 samples, I presented my fingerprinting method to the International Medicinal Mushroom Conference. And what I've been able to do with this method is I've been able to actually come up with a baseline for the different mushrooms, medicinal mushrooms, especially the ones that we sell and the most popular medicinal mushrooms out there. So the baseline for all of our extract products are the actual dried mushroom. So now we use this for all of our different products. We have a fingerprint for chaga, cordyceps, lion's mane, reishi, tremella. For identity, we use DNA analysis, and this is an example of a reishi DNA analysis with the sequences there. And then if for those of you who'd like more information about our method, please pick up my study, Redefining Medicinal Mushrooms, which is here in the booth. Thank you.